I'm delighted to introduce the first speaker of the lightning talks, that is Ingrid Hansson. So Ingrid is a senior lecturer in bacteriology in the veterinary medicine at SLU in Sweden, and her research has been focusing on the identification of sources and risk factors for the colonization of Campylobacter, both at farm and slaughter level. And today she's going to talk about survival on eggshells. I'm really looking forward to the talk because I don't know anything about that. So if you can switch presenters mode, Ingrid.
So,
well, yeah,
like
perfect.
This? Okay, thank you. Okay, thank you very much. First, I want to say thank you to organizers for giving me the opportunity to present our research. I think that most of you know that many animals carry Campylobacter in their intestines without showing any symptoms of disease, and poultry could be colonized with Campylobacter up to about log 8 C4 per gram feces. And as poultry has Campylobacter in the feces, it could also contaminate the eggs during laying. The background of this project is that in the autumn 2018, there was an increased prevalence of Campylobacter in broilers and also in human cases in Sweden. In the autumn in Sweden, we had almost zero Campylobacter in broilers, but something has happened. And we did some whole genome sequencing, and we actually found that some of those flocks actually have the same, eight broiler producers have the same sequence types, although there's not connection to each other. The only connection was that they actually got the broilers from the same hatchery. So we go back and look at the hatchery, and actually we found the same sequence type, ST148, in the same parent flocks as we have for a couple of the producers. And also at the slaughterhouse, the slaughter of these broilers, the workers get Campylobacteriosis. So our question was, could it actually Campylobacter survive outside on the eggs? Is that possible? Could that Campylobacter survive outside and then transport and contaminate the chickens during the hatcheries? And if they survive, for how long time could the Campylobacter survive outside the eggs? So we can see if it was any possibility for the transmission of Campylobacter from parent birds to the chickens. So what we did actually, that we'd have our eggs and they dip them in broth with Campylobacter, and we actually use 3 different serotypes and 3 different concentration of Campylobacter jejuni. And some of those we use Campylobacter, broth with cecum contents, and some we use only broths without cecum contents. And the different sequence types that we used was ST148, the same isolate that they actually found in the parent flocks. We also used ST257 that we found, sorry about that, that we've had, now it's going to, so that we actually found in the water pipes that in one of the broiler producers. And we use ST918 that was isolated from the transport crates after they were cleaned. So one theory, we actually chose sequence type that they thought could actually survive outside in an aerobic atmosphere. So we found an environment because we thought that those sequence type have more ability to survive on the eggshells, since they've actually been isolated from environment. The eggs were dipped in this broth, and afterwards they were separate in laying on these eggshells, in the egg cartons. The first day they were actually kept in a room temperature, and thereafter they were moved to a refrigerator. and analysis was done according the ISO method, just as normal. What you actually found that the eggs could survive, and more than 50% of eggs that have a high concentration of Campylobacter, we actually found Campylobacter 1 day after they was dipped in, after they were kept in room temperature. And then we have some special sequence type 918, where we actually found that they survive also in the medium concentration. What was really amazing was that 10 days after they've been stored in a refrigerator, we actually found this ST918 on the eggshells. That was really amazing results. So our conclusion were that environmental transmission may occur to chicken from eggs contaminated with Campylobacter. The survival, however, differ between the different sequence type. And I think that this risk is bigger if the time between laying and hatching is short, because during this time in that autumn, we have the demand of newly hatched chickens was really high. So there was a short storage time between the laying and the hatching. And then we can have another problem is that could it be a risk for consumer of being effective by the Campylobacter by the eggs? So finally, I want to thank you for your attention. It's a little bit difficult to just have a talk for 5 minutes. And if you have some question, you can please send some questions to me. And I want to acknowledge Jeremy, who was a master student that actually did all this dipping and analysis of the egg. And then my colleagues, Desiree, Sarah, and Liselott. And also, of course, the foundation of Swedish Farmers Foundation for Agricultural Research that financed this study that was a part of a bigger studies. Thank you so much.
Thanks a lot, Ingrid. That was a really interesting talk. I certainly learned a lot in 5 minutes. So we have time for, so we'll take one question per talk. So, and then you can answer the rest on Discord. So we have a question from Tag. Really interesting talk. Was the sample found to be positive after 10 days by qPCR alone or by culture too?
Only culture. We didn't actually have money to do qPCR on all samples. And I was really sorry for that because I would like to have qPCR on all samples. So, but I think that we have found more positive samples if we found the qPCR in all samples, but it was actually culture.
Thanks, Ingrid. Let's move to the next speaker now. So the next speaker is Ruby Coates. Apologies if I don't pronounce the last name properly. She's a final year PhD student at Andrew Grant's group at the University of Cambridge. And yeah, her PhD is on developing a CRISPR-based gene expression tool in C. jejuni. And let's see what she has to say about it.
Hi, thanks for having us to speak. So yeah, my PhD has been working on establishing a CRISPR interference system in Campylobacter jejuni. And as we all know in Campy, there are still genes that we know about that are essential, but we don't know their function. And currently, the molecular tools available in Campylobacter jejuni are quite, there's not very many of them to try and selectively repress genes of interest, because obviously if you knock out an essential gene, you're going to have a lethal phenotype. So the aim is to be able to have a tool to selectively repress genes of interest, so then we can perhaps elucidate their function. So that's where I came in terms of trying to establish this CRISPR-I system in Campy. And for those of you who aren't as familiar with CRISPR-I, basically it uses your traditional CRISPR-Cas9 system has a Cas9 that's guided to your target location with your guide RNA, and that typically elicits a double-stranded break in your target region. The way that CRISPR-I is different from that is that it uses a catalytically inactive Cas9. So what you get is localization to your target gene of interest, but instead of a cleavage, you get a transcriptional block, so you get a gene repression. And this can be controlled using an inducible promoter. So basically, you can induce the amount of ZCAS9 that you flood into this system to regulate the amount of repression that you're eliciting. And this is something that we have been working on in the lab, but due to the short nature of this talk, I'm just going to be talking about my basically proof of concept using the technology in a constitutive sense. So in terms of the key steps to establish this in Campylobacter, it took multiple sort of attempts, but we did a bit of a promoter screen to try and get an understanding as to the level of expression we wanted for our CRISPR machinery. We then selected a range of genes to target and designed sgRNAs against those. And then it was a case of wrapping it all up together and transforming it into jejuni. And I worked with M1CAM and 81176. To show a proof of proof of function of the system, we selected a range of genes. So we selected some biosynthetic genes that are well known in the field, so ASTA and Hippo, and these are really nice because they have a really clear phenotypic readout through various release assays, and you can basically track them using the spec. And we also challenged ourselves and targeted some flagella genes as well. So we targeted FLIGR, FLAR-A, and FLAR-B, which all of you probably know are really important in the flagella assembly pathway, with FLIGR being a major response regulator and driving class-2 gene expression, and FLAR-A and FLAR-B further down the pathway forming the flagellum. So what did we get when we targeted these genes with our CRISPR system? So firstly, in ASTA, we used a large range of guides, and we found that there was a significant repression no matter what guide we used in terms of location along the gene. And this was the case for Hippo as well, although we did see a greater repression at the 5' end of the gene, which is to be expected within CRISPR studies. For FLIGR, we didn't get as much of a resounding success. We didn't get abolition of motility, but we did get a significant decrease in motility. And we're following this sort of qPCR to see exactly what's going on. With FLAR-A as well, we again got a significant reduction in motility, although again we didn't get an abolition, so there might be redundancy in the system, or the CRISPR components might not be expressing to the extent that we need them to suppress these genes, so we're digging into that further. And something cool we did as well, because of the complementarity between FLAR-A and FLAR-B, we actually designed guides that targeted both FLAR-A and FLAR-B at the same time. So we did that as well. And again, we got a significant reduction in motility, albeit not complete abolition. So we'll be doing some analysis into that as well. We'll also be doing some SEM to see what the flagella look like in these strains. So overall, we managed to develop this CRISPR system in C. jejuni and validated it in 2 different strains using a variety of genes. We're now going on to looking at other genes to try and repress, and we have attempts to try and integrate this into an inducible system as well, so we can try and titrate the amount of gene repression that we're trying to elicit. So I'd like to thank everyone in my lab as I'm coming towards the end of my PhD, and thank you all for listening, and I'll be really happy to take questions.
Thank you so much, Ruby. Let me check Discord. Okay, there is no question, so I can ask something a bit more generic. So this is actually the first report on a CRISPR-based interference system in C. jejuni. Is that correct?
That we know of, yeah.
Yeah. So what's you mentioned further targeting further genes. So what's next? What are the next genes in line to be targeted?
Well, obviously, so I sort of started off by saying that we want to be able to target essential genes. So we have been thinking about potentially targeting some essential genes so we can dig into their function a bit more. But obviously, we need to be able to sort of control the system a little bit better so that we don't get full repression and then a lethal phenotype. But that would be hopefully the next step with this technology, yeah.
Perfect. Thanks a lot. You can follow up a question from Martin on Discord about compatibility with other C. jejuni strains there.
Compatibility with other. Oh, okay. Shall I answer that separately?
Yeah, you can answer it here. Yeah, you can do that quickly.
So compatibility with other strains. I mean, so we tried it in 2 strains. It's yet to be known how it will work in other strains, but we're hopeful that it should be able to work within other strains.
Thanks a lot, Ruby.
Thank you.
Right, okay, let's move to the next speaker. So the next speaker is Gillian Carney. So it's a final year PhD student. Are you here?
Yeah,
Gillian?
hi.
Hi. So Gillian is a final year PhD student at the University College in Dublin. And she's working on fluoroquinolone resistance in Campylobacter and its possible impact on the survival and virulence. So the floor is yours, Gillian.
Okay, perfect. Hi, everyone. So yeah, I'm just going to be showing you a little bit of my work on fluoroquinolone resistance in Campylobacter and how we think it could be impacting survival and virulence. So as we all know, Campylobacter is the leading cause of gastroenteritis worldwide, and it most commonly occurs following consumption of contaminated chicken meat. But what's unclear is how it actually manages to survive in the harsh aerobic environment. Another major concern associated with Campylobacter is the rising level of fluoroquinolone resistance that a lot of people have mentioned throughout this meeting. And this has gained this pathogen a priority high ranking from the WHO for research and development. So my project has largely stemmed from the previous work of a member of our lab, Matt Whelan, in which he looked at how fluoroquinolone resistance can impact some key phenotypes. And at this stage, he was interested in this because the causative mutation for fluoroquinolone resistance occurs in an enzyme which can regulate DNA supercoiling. And our lab had already seen how DNA supercoiling can impact a whole range of key phenotypes in Campylobacter. So in Matt's study, he showed that the acquisition of fluoroquinolone resistance due to this mutation can lead to an increase in invasion in the isolates in comparison to the wild type, and also an increase in biofilm formation under aerobic stress. A key aspect of this study, however, was that it was done using a lab-generated fluoroquinolone resistant mutant, so that they took the 1168 strain, grew it in the presence of antibiotics, and created this whole panel where they could compare the wild type and the mutants to see the changes due to the mutation of the gyre enzyme. However, the next question would be then, would this stand true when you look at fresh isolates, comparing ones which are sensitive or resistant to fluoroquinolone resistance? And that's where my project comes in. So my panel consists of some reference lab strains and then also the abattoir chicken strains. And these are ones which are isolated at the abattoirs at the stage of slaughter. Supermarket chicken strains, which are ones which are isolated off commercially bought chicken, just straight off the shelves. And then also human isolates, which are isolates which have been isolated from patients which have been admitted to hospital with a case of Campylobacter infection. So the first thing we looked at is looking at the distribution of fluoroquinolone resistance in the panel. And as you can see, there's been a very high level of resistance across the isolate panel, but the standout group is definitely the supermarket chicken isolates, in that 94% of isolates were found to be resistant to fluoroquinolone antibiotics. And also they have quite a high tolerance. As you can see here, their MIC50 value is up to 50 micrograms per mil. So you just need a lot more of the antibiotics to inhibit the growth of these isolates. Then, as we were already interested in aerobic tolerance and survival in the in a supermarket environment, I also looked at their tolerance to oxygen by just using a simple plate assay. So I just streaked them out on plates and then grew them in the presence of oxygen and then ranked them as either intolerant, low, or medium or high tolerant to oxygen based on their level of growth. And when you compare this data with also the level of resistance that was seen for the panel, you can actually see something quite interesting, that all the isolates which are either low or medium tolerance have some degree of resistance to fluoroquinolone antibiotics, whereas all the isolates which are intolerant to oxygen are also sensitive to fluoroquinolones, which shows that there could be a correlation between these two phenotypes. And since we had the panel with the mix of isolate sources, we also wanted to see if isolate source could impact some key phenotypes. And you can see that they can for certain phenotypes. So in the growth assay, we have here at 48 hours, the OD600 values shows that, especially for the human isolates, they stand out, as at 37 degrees, they have a decreased growth in comparison to the chicken groups, whereas at 42 degrees, they're actually more increased, but probably a lot more variable. Whereas in the motility assay, you can see at 48 hours again, at 37 degrees, the supermarket isolates show the highest level of motility, but at 42 degrees, the abattoir chicken isolates almost catch up to them, but again are much more variable at this 42-degree mark. I also looked at an indicator of the possible virulence of the panel using a cell association assay. So this was just that I labeled the bacteria with a fluorescent antibody and then carried out a 3-hour invasion and imaged them and calculated the average number of bacterial cells per H229 cell, just to get an indication of all the cells which had either adhered or invaded to the cells. There was a lot of variation in the capability of all of my isolates, and you can see that the supermarket isolates are the highest, but this is not statistically significant. So in summary, if I can get to move, yep. I found that isolate source can impact key phenotypes, so that the supermarket isolates do have the highest level of cell association. There's a really high level of fluoroquinolone resistance within the supermarket isolate group, and this resistance may be associated with aerobic tolerance, and we have actually expanded this panel up to a panel of 150 strains, and this holds true in that panel as well. So we think that maybe the supermarket environment could be leading to the enrichment of fluoroquinolone resistance within the Campylobacter population. And thank you very much for your attention.
Thanks a lot. I've been told I pronounced your name wrong, so can you pronounce it for me?
It's Gillian, like it's spelled with a J instead of a G.
Gillian, yeah. Thanks a lot, and apologies for my ignorance on how to pronounce all the names. So we have lots of questions on Discord. I'm just going to take the first one. So from Desi, super cool topic, Gillian. You mentioned that you have created a fluoroquinolone resistant isolate. Did you make this by mutating GRA86 position turn into isoleucine? And which clonal complex were these strains belonging to?
So I personally haven't made any fluoroquinolone resistant isolates. That was the work carried out previous to me. And he just grew them in the presence of the antibiotics and then selected the colonies that formed then. So it wasn't a directed mutation that I think they're describing. And we haven't got the clonal complexes for these isolates yet, but all of these ones have been genotyped at the moment. So we're still waiting on that data. Thanks
a lot. There are some more questions from Discord. So if you could follow up the discussion there, that would be great.
Yeah,
thanks a lot.
Very much.
Right, so let's move to the next speaker. And that is Noemi Tejera. She's a researcher at the Quadram Institute. Yeah, I got this right.
Yes.
She works on the development of a genome-scale metabolic model for Campylobacter.
So good morning, everyone. My name is Noemi Tejera. I work in the Quadram Institute Bioscience in Norwich, UK. And today I'm presenting the differences we have observed in amino acid utilization between two strains of Campylobacter jejuni using HILIC-MS and Genome-Scale metabolic modeling. So understanding the importance of the metabolic capability of Campylobacter jejuni and the differences in substrate utilization between isolates is key in defining how this organism is able to survive and adapt to a variety of environments. In this regard, amino acids play a central role in Campylobacter metabolism, acting as major carbon and energy sources for in vitro growth. So in our group, we use genome-scale metabolic modeling and experimental validation to try to understand better how Campylobacter survive in the food chain. My colleague Dipali is responsible for building these in silico models, which describe the networks of biochemical reactions of a given microorganism. And in-house, we have set up a previous published HILIC-MS method to analyze amino acids in order to investigate how Campylobacter use these substrates and also to validate and curate these in silico models. So initially, we used this HILIC-MS method to analyze the amino acid consumption of M1 after 24 hours growing in a modification of a commercially defined media. So it was nice to see that most of the amino acids that are clearly preferred by M1 had been identified as auxotropic or growth promoting in a study that we published last year. So to dig a bit deeper into these preferences, we set up an experiment that we see here, where I culture M1 and another C. jejuni strain, 1116AH, using the same modified DMEM media. And I collected samples at different time points to analyze both growth and also the amino acids consumption. So these are the LC-MS results. In each graph, you can see the consumption of amino acid expressed as percentage against time, and also the initial concentrations for each amino acid. I have highlighted in green those amino acids that show the highest consumption percentages. So when we look here, we can see the growth curves for this is M1 and this is 11168H for this experiment. And I have put in these circles when 100% utilization was reached for each amino acid and for each strain, in green for M1 and in blue for 11168H. So aspartic acid and serine were the first amino acid to be completely catabolized by both strains. This is something that has been well documented before, and it was nice to see that this is the case, and also match the results that we obtained with our metabolic models. Proline is also an amino acid that is preferred by Campylobacter, and we can see how clearly it is consumed very early on by both strains. Cysteine, according to the literature available, is considered isotropic for 11168H, and our model gave us the same result. And from the LC-MS, it's nice to see here how the consumption of this amino acid goes in line with the growth of this strain. For asparagine, this amino acid was an interesting one because we can see how it's clearly consumed completely very early on by M1, but not by 11168, sorry. And this could be a reflection of how effectively each strain transformed this amino acid into aspartic acid, that is the one that is entering the TCA cycle. Next, we have methionine, which we have shown in our previous published study that is isotropic for M1. And again, we can see how the consumption of this sulfur-containing amino acid goes hand-to-hand with the growth of this strain. Glutamate is a very well-known preferred amino acid for M1, and it was very nice to see the presentation from Ashley Griffin talking about glutamine, because we see that in presence of pyruvate, in our media we have pyruvate, is well consumed in our experiment. And these amino acids, leucine, isoleucine, phenylalanine, and tyrosine, were very interesting to look at least for M1. And I can say that including them in the minimal media that we published last year improved the growth of both strains at 24 hours. So with this, I would like to thank my supervisor, John Wayne, my colleagues, Deep Allison and Mark Philo, our collaborators, and of course, the funding body for our research. And thank you all for listening.
Muchas gracias, Noemi. I don't see any questions in Discord, so I can ask a quick one myself. So these two strains that you're testing, and you see the differences in the utilization, can you just elaborate a bit based on their strain ecology, so to say? What could they mean?
Well, correct me if I'm wrong, because this is not my field, but I'm pretty sure that 11168H is in clade 3 and M1 is in clade 9, I believe. So they are like both from human origin, but they're in separate clades. And the reason why we use them was basically because we collaborate with the London School of Hygiene Tropical Medicine and Cambridge University, and we wanted to be able to compare results and work in each other's research. So it was a collaborative way to do it.
Thanks a lot, Noemi. You can follow up if there are any questions from Discord.
Yes, sorry, stop check. Perfect, thank you.
Great. Let's move to the last two 5-minute talk presentations. They are both recorded. So the first one is Anja Klančnik. I probably pronounced that wrong. She's an associate professor at the University of Ljubljana, and she's focusing on novel targets to combat Campy. looking at resistance, stress, and different stages of biofilm. So please, if you could start the talk.
I'm Anja Klančnik from University of Ljubljana Biotechnical Faculty. Thank you to the organizer for this opportunity. I'll present the work of Slovenian Campi group. The main purpose of this story is to control this pathogen, which is one of the most important goals for ensuring food safety and public health. This is how microorganisms work together within the biofilm. The occurrence of persistent condition is a factor in the survival and spread of resistance, as well as source of contamination, and also a starting point for further infection and colonization. The formation of biofilm allows the adhesion of Campylobacter. The study of this interaction would reveal the main mechanism that could consequently be prevented. The adhesion is very complex and insufficiently researched, so we approach this holistically on several levels. First, we studied the adhesion to various materials of production surfaces used in food industry, such as steel and glass, and to biotic surfaces, such as human cell line and eukaryotic amoeba cells. In addition, challenge is to use the appropriate method or introduce new for evaluation of adherent and biofilm cells. We used indirect quantification based on determination of biomass, culturability, survival, metabolic activity, aggregation, and morphology. We also introduced modern method as direct quantification approach using real-time quantitative PCR or digital PCR that detect and quantify low level of DNA, which are very sensitive and specific methods. Thus, we can focus to the basic mechanism of adhesion and biofilm formation. After the first phase of adhesion to polystyrene in the first 12 hours, the number of cells increases to 24 hours. Then the number of cells in the formed biofilm decreased. We also evaluated the adhesion in correlation with the interaction of microbes on the surface of material common in food processing and on two biotic cell models. The fact that Campylobacter in co-culture with other pathogens increase adhesion highlights the problem of microbial aggregation. We also compared the morphology of Campylobacter adhered to the abiotic and biotic surface under the electron microscope. At high magnification, interaction through some structure were seen in both monoculture and co-culture with other pathogens. Further, we studied which cellular mechanisms could present a new target, the role of a flux system, intracellular signaling, membrane capsular system, mechanisms of general stress response, and motility in the adhesion was evaluated using mutant strains. Reduced adhesion was associated with a flux pump, extracellular proteins, signaling, interaction, and stress response. Subsequently, we introduced the adhesion approach of Campylobacter after the addition of antimicrobial agents of various origin, but in their subinhibitory concentration. As we study at transcriptomic level, variety of adaptation mechanisms, including stress response and metabolic modulation, were used. Last but not least, we focus on the study of unexplored extracellular matrix molecules involved in adhesion. Due to specific properties of carbohydrate recognition, we use lectins as kind of biosensor to investigate non-cellular molecules involved in adhesion. Thus, we gain some knowledge of Campylobacter adhesion at physiological and genetical level. In summary, Campylobacter adhesion is greater in interaction with other microorganisms. In Campylobacter, cell adhesion modulates other cell properties and triggers stress response, modulation of metabolism, motility, and other variant factors. Knowledge of the metabolism allows inhibition by adhesion, but important is also the fact that anti-adhesion has proven to be a very successful model of study. By reducing the adhesion, we consequently control the contamination. I would like to thank the Slovenian Research Agency and invite you also to focus on other presentation of our group. Thank you for your attention.
Thanks a lot, Anja. I don't know if you're in the chat.
Yes, I'm here.
I don't see any questions on Discord, so I can ask one myself. So you're focusing on 11168 to look at different assays. Is that correct? The biofilm, adhesion.
Yes, we are looking.
So we saw in a publication of ours with in collaboration with Dave Kelly that there is variation within different strains that have, so to say, different ecology in Campylobacter. So the chrono complex 21 and the chrono complex 61, which is found mostly in cows. So are you planning to have a look on how different clones of different ecologies, so to say, act when you put them into different assays? Because we saw different differences in biofilm, for example, when we compared strains isolated from chickens compared to strains isolated from cows.
Yes, thank you. This is a good suggestions. Each strain has different biofilm formation. This is the variety between the strains. So if the strain is food or even animal, this is really big difference also on physiological level and also on genetics. So I think that these are also influenced also to biofilm and adhesion processes. So yeah, it's good to research different strains, but it takes a really lot of time.
Yeah, I figured that. Okay, I don't know if anyone has anything on the Discord, but you can follow anything there. Thanks a lot for the presentation. And let's move to the last speaker for today. So it's another recorded talk. It's from Morgan Menning. She's final year PhD student at the National Health Laboratory in Luxembourg and works on recurring genotypes of Campylobacter jejuni involved in human infection and associated to specific phenotypic traits. So let's listen what she has to say.
Hello everyone, my name is Morgan and I am a PhD student at the National Public Health Laboratory in Luxembourg, and I work in collaboration with INRAE, a French institution. First, I would like to thank the organizing team to give me the opportunity to present some of my most interesting findings. I am going to talk about the investigation on recurring genotypes of Campylobacter jejuni involved in human infection and associated to specific phenotypic traits. Campylobacter emerged as the leading cause of bacterial gastroenteritis worldwide. Since 2006, an increased trend for human campylobacteriosis has been observed in Luxembourg, which motivated the monitoring of the circulating genetic profiles. Isolated from diverse sources such as human, food, veterinary, and environment were collected in routine for molecular typing purposes. In our previous study, by comparing of genomic profiles, we discovered that almost half of the patients were infected by recurring genotypes in Luxembourg. Regardless of the typing scheme used, the results support the clonal expansion of stable genomes in Campylobacter populations. Persistent strains may have acquired specific phenotypic traits to adapt to other hosts or disperse in the environment. That's why we perform different tests on a strain collection. These strains were isolated between 2005 and 2018 from multi-host and with recurrent or sporadic profiles. Whole genome sequencing data were analyzed with a core genome-MLS scheme composed of 1,343 loci. This scheme was then implemented in Sexio+ in order to perform a phylogenetic analysis. You can see here the 4 recurrent lineages, with 2 defined as host generalist and 2 as host specialist. The sporadic strains are displayed in red and blue according to their sources. So the aim of this study was to investigate if recurrent strains could adapt to aerobic conditions, could survive oxidative stresses, could adhere to an innate surface, and could develop biofilms. These specific phenotypical traits were previously shown to contribute to persistence in the environment and human transmission. That's why we investigated the collection presented previously. Concerning the results, of the 84 strains to date, 45 and 31 strains were able to survive at a concentration above 0.25 millimolar per quart and H2O2. we demonstrated that responses to superoxide and hyperoxide stresses are correlated. When a strain survives to a concentration of 0.25 millimolar per quart, it is 6 times more likely to survive to H2O2 at the same concentration. In addition, recurrent lineages have developed more phenotypic adaptation. Lineages A and B, here and here, survive oxidative stresses significantly more than lineage C and D. No particular phenotype could be correlated to a genotype for the sporadic strain. Concerning the adhesion capacity and the biofilm formation, you can see here that a high adhesion capacity was measured for 39 strains, and 27 strains developed a biofilm. We also demonstrated that adhesion capacity is mainly correlated to biofilm formation. When a strain has a strong adhesion, it is 14 times more likely to develop a biofilm. And concerning the recurring lineages, you can see that lineage G has the highest capacity for adhesion and biofilm formation. Concerning the acclimation to aerobic condition, unexpectedly, of the 84 strains studied, half of the strains could acclimate to aerobic condition. No correlation was found between the acclimation of the strains to aerobic conditions and their survival to oxidative stresses, their adhesion capacity, their biofilm production, or their genotype. The molecular mechanisms for adaptation to aerobiosis and for survival to oxidative stresses would therefore be different. So in summary, we highlighted a good correlation between phenotype and genotype, and each recurrent lineage is characterized by a specific phenotype. This data suggests that phenotypic adaptation may contribute to the persistence of stable clones over time and from different sources. That could explain a better transmission to humans and representing a public health risk. Thank you very much for your attention, and if you have any question, feel free to contact me.
Thank you so much, Morgan. I don't know if there are any questions on Discord. I can ask one. Can you go back to the slide where you had the isolates and the clonal complexes?
So you have a question?
Yeah, I just wanted to see which were the clonal complexes that you tested the different phenotypes, and it's just a thought that it would be nice to include some extra strains that they are isolated from the environment, and they belong to a different sequence type that's not associated with agricultural animals. Those I suspect will be the ones that will do good.
So I have some sporadic strain from human, from your environment, and I have also in each lineage from multi-host. So I have from poultry, from bovine, and human also.
Yeah, I was
water also. Sorry,
it's just a thought of including in the analysis or in your assays isolates that they are not linked to agricultural animals. So isolates that are isolated from water, for example, and they don't belong to any of these known. You included those in the analysis?
Yeah.
Okay. And those, there was no correlation between the genotype and the phenotype for those ones?
For the sporadic one, no, not really.
Okay. Good. Well, I have nothing else to ask. So that was the end of the session. Thanks a lot, Morgan, for the talk.
Yes. Sorry, I don't find how to stop to share. Oh yeah. Good.
Great. So thank you very much to all of our speakers for the excellent talks in the session today. We've had varied and very enjoyable talks. It's great to see all the interaction on Discord. So do please keep that up. Obviously, I would like to extend a big thank you and well done to Evangelos for chairing this session today. A fantastic job. So well done, Evangelos. So coming up at 1 o'clock, we have the industry session. So we look forward to seeing as many of you there as you can. We've got two poultry companies coming along to have a chat with us. There's a Discord channel if you would like to propose any questions that we can put to the panel members. And then, of course, we have the plenary panel at 4 p.m. today. There's also a Discord channel for that one. So if you have any questions that you would like to ask to either the industry representatives or the plenary panel, do please use the Discord channels for those. Finally, after the plenary panel, we have a social event planned. This is going to be happening via the Discord platform, where people can meet and talk. We're going to be running a pub quiz with questions about Campylobacter and similar topics. So if you're interested in participating in the quiz, please let us know on the pub channel in Discord. So thank you very much, everybody. Thank you for your attendance at the session, and we will see you soon.